Hi everyone, so this is the sixth mini lecture of chapter 13. Hopefully these have been helpful. I do find I talk with my hands when I do this, so you guys are missing lots of good hand movements. Anyway, this is the last one on chapter 13. The learning outcomes for this one are looking at different immune states, and then we're going to talk about vaccines, what makes an effective vaccine and how we categorize them, and then why vaccines are so important, and especially when we're talking about a herd immunity. So we're going to start with specific immunity and vaccination. There's two ways that we can acquire immunity. The first would be a natural immunity, and this would be through a normal biological experience. For example, getting the chickenpox from your sibling. We also have artificial immunity, and this is protection from an infection, and it's obtained through medical procedures. So often this would be vaccines. There are differences, though, that we can have under both of these. We can have either an active or a passive immunity. Active means that the individual receives immune stimulus that will activate their B and T cells. So in this case, we're actually activating the immune system. And in this way, you would create memory for quick action in case you're re-exposed to that antigen again. It takes several days, though, to develop, which is one of the downsides, right? Because your immune system has to ramp up. But it will last for a relatively long time, and it can be stimulated either by natural, for example, an infection, or artificial means, and this would be a vaccine perhaps. We can also get passive immunity, and this can be that individuals receive antibodies from another human or animal. So they're not actually making the antibodies, but they are given antibodies that will work against a particular antigen. In this case, the recipient of those antibodies will only be protected for a short period of time, even though they haven't had any exposure to the antigen before. In this case, it would lack memory because we would not be stimulating our own immune system, and we would not be able to produce our own antibodies against that disease. And for that reason, it has a very short-term effectiveness, but it will have immediate onset of protection. And this can be done either naturally or artificial in origin, and we'll talk about that on the next slide. So on the top box here in the blue, these are all natural ways that you might acquire immunity. The first is active, and this is talking about recovery from infectious disease. And that's kind of going through those immune stimulations that we saw with B and T cells. It is possible to get passive immunity naturally, and in this case, this would be a result of prenatal and postnatal mother-child relationship. So perhaps things that were passed through the placenta or through breastfeeding. But this would be a way that an infant could get immunity without actually producing their own. And this is helpful, especially very early on in life, in those critical months before their own immune system has really started working. Now, we can also do artificial immunity. The first type we would say is active, and this is where we have a vaccination that you're exposed to a particular antigen, and then your own immune system is what gets ramped up, stimulated, and protects you and also forms a memory. We can have passive immunotherapy, and in this case it would be specific antibodies against perhaps a particular infectious disease. So you would get it from a donor, somebody who had been exposed and had produced antibodies, and then the antibodies would be put in you to fight off an infection. In this case, you would not maintain that immunity after those antibodies left your system. So when we look at artificial active immunity, we're really talking about vaccination. And when we think about vaccination, what we really want to do is we want to stimulate a primary response and a memory response. So we want to stimulate the B and T cells into action, but also have them put some memory cells tucked away in case we see that antigen again. So they get that immune system ready in case we get exposure to a virulent or a pathogen that can cause damage. And then this way, if that pathogen eventually enters the body, the response of your immune system would be very fast, and it would be very strong, and it would last for a period of time to get rid of the infectious agent. So what are the principles about vaccine preparation? What do we look for in an effective vaccine? The first, obviously, is that we protect against exposure to the natural or wild forms of the pathogen, so what you would be exposed to in your daily life. We want one that's safe with very few adverse effects, and we want one that can stimulate both our antibody or B cell response and our cell-mediated or T cell response. So you want to have both of those parts of your specific immunity stimulated. In addition, we want it to be long-lasting, right? We want it to produce a memory so that later on down the road when you're exposed to the pathogen, you have protection. It's helpful if it only needs one dose, or at least not very many doses or boosters. It's hard to get people in to take more than the initial or maybe first couple of shots. And we want it to be inexpensive. We want it to be something that we don't have to pay a lot of money for, that it can last a while at a doctor's office, and that it's easy to administer. This is especially true when we start looking in countries that are developing, and we need it to be easy to get to these people. So what are the different types of vaccine preparations? We have whole cell or virus vaccines, and in this case we have one or two ways that we can go. We can have live attenuated cells or viruses. So this means that viruses, or if it's a bacteria, that they're alive but they've been damaged in a way that they cannot cause an infection. And then we can have killed cells or inactivated viruses, and in this case these are still maintain their antigens, but they cannot reproduce, and we'll talk about that in a minute. And then the last thing that we could have is we could have antigenic molecules. So if we know what it is from a bacterial cell or virus, the actual subunit that can cause an immune response, we may be able to manufacture these, and then we probably have to put them in conjunction with another protein so that they're large enough to get that immune response. And these would be called conjugated vaccines, meaning that we have taken those subunits and we've added proteins so that they create a better stimulus of the immune system. So this table is a really great one showing the different types of vaccines. At the top we have those whole cell vaccines, and at the bottom we have the subunit vaccines. So you can see here we have those killed or inactivated viruses. So basically either heat or chemicals has been used on either the bacteria or the virus. They're dead, but their antigens are still able to be retained so that when they're administered to the host, it will stimulate an immunity, but the pathogen cannot multiply. So this is one way that it can be done. The other way that we can do this is that we can use what we would call live attenuated cells. So either the virulence of it is eliminated or it's reduced to the point where it will have a very mild response. And in this case, it's alive and it has the antigens that it would have in its virulent state, but it's not going to cause as big of a problem. When this is administered, these can actually multiply, which will actually boost the immune stimulation in comparison to dead ones, because you'll have a number of different microbes that are being reproduced. So the advantages of that live preparation that I was just talking about is that they can multiply, which means that they can cause an infection, but not very badly, and so the immune response will be very strong. They will confer a much longer lasting protection, and they usually require less doses or boosters than the first type. And that's because with that multiplication, you get more exposure to it. And then they're very effective at inducing the cell-mediated immunity, or the T cells. Now, there are disadvantages to the live microbes in vaccines, in that they often require special storage, right? They're going to be live, so you can't just have them on the shelf. And there is a chance that they could mutate back and become virulent again. And so that's something that healthcare workers are already thinking about, and researchers are working on and trying to figure out how to make sure that that doesn't happen. For the most part, we probably get more of the killed cell or inactivated virus as vaccines. And then that last one is the subunit, and here it shows if there were markers perhaps on the virus that we could then modify or replicate on its own and actually use that, or perhaps a different part of the bacteria, a protein or something from the cell membrane or cell wall, or the flagella that we could use. In addition, we can do genetically engineered antigens by placing a foreign microbe antigen within a plasmid, and then it would produce that protein that might stimulate the immune system. Or toxins, we can actually make toxins where we denature it slightly, and that toxin then would be able to make a vaccine. So why do we do vaccines, and what's the reasoning behind it? The problem when you don't do vaccines is that we have outbreaks of communicable diseases that often can lead to death for our most vulnerable and sometimes even our healthy. So without vaccinating, we can have outbreaks of measles, mumps, diphtheria, polio, typhoid fever, and whooping cough. And it's important, even if we're not one of the vulnerable populations, that we do herd immunity, which means that if a certain percentage of the population is vaccinated, it means that that would be impossible for a microbe to circulate. And this is really important when we look at people who have immune suppression or young children that are not old enough to get vaccinated. As long as enough of us, a high enough percentage of the population is vaccinated, there is protection for those ones that cannot be vaccinated. And so this is a really important part of vaccinations. So getting vaccinated not only serves that common good of protecting those that can't be vaccinated, but it also will serve the individual good, because the person who gets the vaccine will not get the infection. So before we had the measles vaccine, just as an example, we had 3 to 4 million cases of measles per year, which is a lot when you think about that. And out of those, we had between 300 to 400 children died annually from measles. So this is completely preventable using a vaccine. And you can imagine that, how huge that would be when you look at the population of the US. And then 1,000 of those that were infected out of that 3 to 4 million cases of measles a year, over 1,000 of those were chronically disabled due to measles encephalitis, which is a swelling of the brain. So what they've kind of shown now is that childhood vaccines can save the lives of up to 2.5 million children a year worldwide. So it's very helpful, and one of the things that we've learned through microbiology to protect others. So checking in, you should know the active and passive immune states, both natural and artificial. What category of vaccines do the microbes replicate? What category of vaccines are longer lasting? And then be able to explain what herd immunity is, define it, and why it's important in a given population. So that should conclude my Chapter 13 mini lectures. I hope these are helpful, and I hope you guys have a great Thanksgiving break. We'll see you after the break. Thanks.